Welcome students. Kindly subscribe my channel. Today, actually this is for pharmacognosy and phytopharmaceuticals for second Form D students. It is very important actually. The chapter is small, the last chapter. There are many other chapters to be covered. If it is left, you have to inform me. This especially proteins. I would like to inform the students not to follow the printed notes or the old notes which is being used again and again. Don't. You prepare your own notes by listening to my lecture. Kindly do this. I am repeatedly informing the students of all the sessions. Kindly prepare your own notes. By listening, you can make hints, and also the technology giving more advantage. You can pass it, you can write, you can go ahead. So this you can utilize it. You prepare your own notes, you will succeed. It's not necessary even for you to read again and again. Understand? You prepare your own notes. This is actually proteins. I have taken the topic to complete in one lecture, but it will be somewhat a little time I need, because I need to give some the questions. Also we have to discuss what are the questions which they have asked in university exam. So that will be more interesting for the student for the study. I have been repeatedly telling in the classroom also, you have to always have your syllabus with you, and as well as you should know about the previous question papers, what they have asked from the particular chapter is needed for the students to revise, and reading the syllabus again and again. What's the extent I am going for my reading and my preparation? It's needed for the students should know this one. The past Kerala University of Health Science questions from proteins for Form D students. You see, define protein, give the chemical test for the identification of protein. 5-mark question. Define protein and classify with examples. In December 2023, that is October 2024, recently, this is December 2023, 5-mark question. Define and classify protein in March 2023, 5-mark question. Write the source, preparation, test and uses of gelatin in March 2023, 5-mark question. What are proteins? Explain the chemistry and method of analysis. Method of analysis of protein I have discussed in this lecture. Very important. You have to write clearly. In May 2022, 10-mark question. 10-mark question they have asked. Methods are very interesting. Even up to quantitative estimation you have to write. Define and classification. You have to classify and general test for protein. Test for proteins which you know very well about the xanthoprotein test, Bayur test. These all tests I have given in gelatin. So you can write general test. If they ask protein also, you can mention that. In November 2021, 5-mark question. You say, write the source, preparation, test and uses of gelatin in March 2021, 5-mark question. Every year they are asking. Even you see in March again they have asked 2 questions. Gelatin they have asked, and also define and classify protein. She's a 10-mark question in March 2021, same question paper. In January 2021, 5-mark question, define protein, add a note on any one protein containing drug. You can write gelatin, collagen. Chemistry of protein, 5-mark question. December 2019, before Corona, the last exam, they have asked 5-mark question. Write the source, distribution, chemical constituents and uses of gelatin in June 2019. 5-mark question. Write the various chemical tests involved in the identification of gelatin in June 2019. 5-mark question. 2 questions they have asked in the same question paper. Define protein, explain the classification, chemistry, production, chemistry, a chemical test and uses of gelatin, including 10-mark question in June 2018. 10-mark question they have asked. Describe the method of analysis of protein in January 2018. 5-mark question. Classify proteins and add a note on collagen in July 2017. 5-mark question. Distinguish test between starch and gelatin. Carbohydrate, you might have studied the various tests. You can mention all the tolerance test, fillings test, and difference between their chemical nature and physical nature. Compare with gelatin. In January 2017, 5-mark question. Method of detection of protein in July 2015, 5-mark question. So like that, it is repeated question they have asked. So kindly listen to my lecture and make yourself a notes, and it will be more supportive and more helpful for you to appear in your university exam and to give a very clear-cut explanation. Proteins are large complex molecules made of amino acids essential for numerous bodily functions, including structural support, enzymatic reactions, and immune responses. They are the building blocks of tissues and organs, and their unique sequence of amino acids dedicate their specific three-dimensional shape and functions. As shown in the picture, you can see that tertiary structure, primary structure, X-helical, alpha-helical structure, and 2 anti-parallel beta strands with lobe is shown in the beta sheets, can be seen very clearly about the three-dimensional shape function of the protein. Animal source of protein: lean beef, lamb, whale, that means the small cattle, pork, poultry, chicken, turkey, duck, fish and seafoods, fish, prawns, crab, lobster and salmon. Eggs is a complete protein source. Dietary products like milk, yogurt. Plant-based sources for proteins: legumes and beans like lentils, grains, chickpeas, split peas, tofu and black beans. Tofu is condensed soya milk that people press into block of different firmness like this, like a paneer which you prepare from the milk. Nuts and seeds: almond, pine nuts, walnuts, cashew nuts, pumpkin seeds and sunflower seeds. Quinoa, a complete protein source. In Tamil it is called seemai thinai. In Malayalam, quinoa. Even in English also, quinoa, it's quinoa. This is the plant with the grain or the seed, and this is the quinoa seeds. Quinoa is a seed of Chenopodium quinoa plant. So quinoa plant is a very good complete source for protein. Protein powders like whey, casein, soya and pea protein powders. Whey is a protein which is available, is a thin liquid that is left after sour milk. After the solid part, curd, have been removed. Protein functions, it acts as a catalyst. Enzymes or proteins enzymes or proteins that speed up the chemical reaction. Structure, protein provides structure support for cell and tissues. Transport, some proteins carry molecules such as oxygen in hemoglobin. Regulation, proteins can regulate gene expression and cellular process. Defense mechanism, antibodies are proteins that protect the body from pathogens. Classification, very important section we should know about classification. They are asking repeated questions. Write it like classification can be done by its composition. Classification can be done by its structure. Classification can be done by its function. So protein can be classified in three ways. By composition, simple protein, conjugated protein, derived protein. By structure, globular proteins, fibrous proteins. By function, enzymes, structural proteins, maintaining structural proteins, and transport proteins, hormonal proteins, contractile proteins, defensive proteins, storage proteins, and genetic proteins. So by function, this many classification can be given for proteins. So we should remember that the number one by composition, number two by structure, number three by function. In number one, in composition, simple protein, conjugated protein, derived protein. And in number two, by structure, globular protein, fibrous protein. And in number three, function, by function, enzyme and structural proteins, transport proteins, hormonal proteins, contractile proteins, defensive proteins, storage proteins, genetic proteins. So this is very important to remember by the students and write in this way. Classification by composition, individually we should have some small definitions should be written. Simple protein means these proteins upon hydrolysis yield only amino acids. Example include albumin, globulin, histones and protamines. Conjugated proteins, these proteins contain non-amino acid components such as lipids, carbohydrates or metal ions in addition to amino acids. Examples include glycoproteins, lipoproteins and nucleoproteins. Derived proteins, these are formed from simple or conjugated proteins through chemical or physical changes such as denatured proteins and peptides. Classification of protein by structure. First one is we know globular protein, which is spherical in nature. By seeing the picture, these proteins are generally water soluble and have a spherical shape. Example include enzymes, transport proteins, and antibodies. Fibrous proteins, it looks like a thread. These proteins are typically insoluble in water and have a long thread-like structure as shown in the picture. Example include collagen and keratin. Classification by function. Enzymes, we know that. I made it little colored. Very important. There are proteins, enzyme proteins, very interesting therapeutic activities and physiological activity, important enzymes I have mentioned. So it should be remembered individually. You can refer and study also. This protein catalyzes biochemical reactions. Example, papain, which is from papaya, bromelain, which is from pineapple, steatoheptidase is enzyme from kidney tissues of animal, urokinase, streptokinase, they are also from kidney tissues from animals, having more use in blood clots, in critical conditions for patients to be treated in various cardiac issues can also be solved by means of streptokinase. Pepsin, we know that it is a digestive enzyme which is also produced from animal sources, from stomach linings, which is prepared. So we should know that these enzymes classified by its function. Structural proteins, these proteins provide mechanical support and maintain cell shape. Key examples include collagen, keratin, and actin. Transport proteins, these proteins transport molecules across cell membranes or within the body. Examples include the sodium-potassium pump, which transports sodium and potassium ions, and another example, glucose transporters, GLUTs. They help transport glucose into cells. Hormonal proteins, these proteins regulate various biological processes. Example including these hormonal proteins, insulin, glucagon, thyroid stimulating hormone TSH, and growth hormones. Contractile proteins, these proteins enable muscle contraction. Two primary examples are actin and myosin, which work together to facilitate muscle contraction. Defensive proteins, these proteins protect against pathogens. Examples include antibodies, lectins, and antiviral proteins. Storage proteins, these proteins store nutrients. Examples include seed storage proteins like 2S albumin, globulin, and prolamins in plants. And ferritin, which stores iron in animals. Other examples include casein in milk and ovalbumin in egg whites. They act as storage proteins. Genetic proteins, these proteins are involved in the regulation of gene expression. Example include enzymes, structural proteins, hormones, and antibodies, which all we come across in various functional proteins. These proteins are crucial for building and maintaining cells and for carrying out all life process. Gene proteins. Now, what are the physical properties of proteins? Color and taste. Proteins are typically colorless and tasteless. Shape and size, they range from simple crystalloid spherical structures to long fibrillar structures. colloidal nature, proteins, due to their large size, form colloidal solution when dissolved in water. Amphoteric nature, they act as both acid and bases depending on the pH of the solution. Solubility, protein solubility is influenced by factors like pH, ionic strength, and nature of protein itself. They generally have higher solubility at pH values far from their isoelectric point. Denaturation, change in temperature, pH, or exposure to certain chemicals can cause protein to lose their shape and function. A protein, that process is called denaturation. Molecular weight of protein. Proteins have varying molecular weights depending on their number of amino acid they contain. Optical activity. Proteins are optically active, mean they can rotate the plane of polarized light in physical. In chemical, we can talk about the chiral carbon atom. Foam formation. Some proteins have an ability to form stable foams, which can be important for the food process. Emulsification. Protein can also act as an emulsifier, helping to stabilize a mixture of oil and water. Chemical properties of proteins. Neutrality. Proteins generally have an overall neutral charge, as we know that it has carboxylic acid and amine group, COOH group and NH group. So, acid carboxylic group of some amino acid balance the basic chain of the other. Solubility. Protein solubility is influenced by pH, with proteins being least soluble at their isoelectric point, where their charge is 0, and becoming more soluble in acidic or alkaline solutions. Optical activity, again we are talking about in chemistry. As I told, it is chiral carbon atom of the alpha carbon atom. Protein are typically laboratory negative, meaning that rotate the plane polarized light in the left side. This is due to the chiral or chiral, how can you pronounce chiral, nature of the alpha carbon atom in amino acid. Number 4, peptide bond. NHCO or CONH bond. Protein are formed by linking amino acid together with peptide bonds, which are amide linkage, CONH, linkages between the carboxylic group of one amino acid and the amino group of the next one. Denaturation we have discussed already in the physical also. Protein can lose their original structure and the function through denaturation in the process that disrupt the bond that maintained their 3D structure. Denaturation can be caused by heat, increase in temperature, pH changes, radiation, and chemical agents like detergent or organic solvents. Methods of analysis of protein. They have asked question like that, methods of analysis of protein. That's why I made it in such a way, how to analyze the protein. You have a step of extraction, isolation and characterization. The same thing you have to follow. Separation technique. First you have to separate the proteins. Second, identification technique. And the third one is characterization technique. And fourth one is also there, quantitative estimation. So this all analysis clearly you have to specify. In separation technique, electrophoresis, in which SDS phase is a very important chemical electrophoresis, SDS, that is sodium type of gel which can be used in electrophoresis type for separation of proteins. Isoelectric focusing, chromatography, Western blotting. In identification technique, mass spectrometry, Edman degradation, sequence alignment, antibody based methods. Interesting to remember all these identification techniques for proteins. Characterization technique, structural analysis, activity assays, spectroscopy, protein-protein interaction analysis. See the category how separation technique, identification technique and characterization technique. Like that you have to mention the method of analysis of proteins. Finally come to the quantitative analysis of protein, in which Kedal, pronunciation K-J-E-L-H-A-D-A-H-Ls, you can pronounce Kedal's method. Kedal's method is an interesting method for quantitative analysis of protein by using the nitrogen ion, nitrogen. Modified Damas method, UV spectroscopy method. We will discuss that. Very interesting and very simple for you to remember if you note it down the point by yourself by listening to the lecture. Number 1, separation technique in analysis. Number 2 is identification technique. And number 3 is characterization technique. The student should know very clearly, you should mention. In number 1, separation technique, you should mention about electrophoresis, which specifically with an SDS phase electrophoresis can be followed. Isoelectric focusing, chromatography, and the fourth one, Western blotting technique. In identification technique, mass spectrometry, Edman degradation, sequence alignments, and antibody based method. In the third one, characterization technique, structural analysis, activity assays, spectroscopy, protein-protein interaction analysis. And in the fourth one, finally, quantitative analysis, quantitative estimation of protein can be done by Kedal's method, modified Damas method, and UV spectroscopy method. There are many others, but still the student should remember all this very clearly. You can mention point by point. It will be the right answer for analysis of protein. Protein separation techniques. First, by electrophoresis, separation of protein based on size or charge using an electric field. Separately, I have a class about electrophoresis. Even it is possible for the students to know about electrophoresis. You can listen to it. SDS phase is a part, it's a type of electrophoresis, a common technique for separation of protein by size. SDS phase means sodium dodecyl sulfate polyacrylamide. And this gel electrophoresis in a technique used to separate protein based on their size, molecular weight. SDSPAGE means, you should remember, S stands for sodium, D stands for dodecyl, and S stands for sulfate, and P stands for poly. A stands for acrylamide and G stands for gel electrophoresis. So that's what you have to remember. SDS PAGE, that is electrophoresis, is used for protein separation technique. Isoelectric focusing separate protein based on their isoelectric point. Chromatography separates proteins based on various physical properties including size, charge and binding affinity. Western blotting identifies specific proteins from a complex mixture after separation by size. It involves separating proteins, transferring them to a membrane and using antibodies to detect the target protein. Protein identification technique, the second one. Mass spectrometry, measure the mass to charge ratio, line spectra of ionized protein to identify them. Edmond degradation means sequence amino acid in the peptide by integratively removing one amino acid at a time. The Edmond degradation removing in a chemical method using for sequence peptides and proteins by sequentially removing and identifying the amino acid residue at the N terminals. The peptide N terminals can be determined. That is Edmond degradation. Sequence alignment or alignment, you can say. Not alignment, alignment, you can say. Sequence alignment compares protein sequence to database to identify homogeneous proteins. Antibody-based methods utilize antibodies to recognize and identify specific proteins. Protein characterization techniques, in which structural analysis determining the 3D structure of proteins using techniques like X-ray, crystallography, NMR, nuclear magnetic resonance spectroscopy, and cryo-EM, that means cryo electron microscopy, where we can, by means of electron microscopy, the picturized can be easily photographed. Activity assays, assessing the enzymatic activity or other functional properties of a protein. Spectroscopy, in characterization techniques, using various spectroscopic techniques to study protein properties including fluorimetry, that means fluorescence, UV visible spectrometry or spectroscopy, and infrared IR spectroscopy. These all spectroscopy can be used for characterization of protein. Protein-protein interaction analysis, studying how protein interact with each other using techniques like, very important technique, that's why just highlighted by yellow color. Co-amino precipitation, that is COIP, it is mentioned in short form COIP, co-amino precipitation technique, protein-protein interaction analysis, yeast 2 hybrid techniques, and surface plasmon resonance technique. These three techniques can be followed for protein-protein interaction analysis. Now, quantitative analysis of proteins. Interesting how we can go for quantitative analysis. By Kedal's method of quantitative analysis of proteins. You see the Kedal's flask, how he has added an alkali sodium hydroxide after digestion of protein sample and making the nitrogen to be separated as an ammonia and titrated against an acid. Like that you can remember. It involves digestion of sample with sulfuric acid, alkalizing the solution and then titrating the liberated ammonia. Once if you listen, you can understand. Nitrogen is present in the protein due to the building blocks of amino acids. So that nitrogen can be made into liberated ammonia. First step is digestion. The sample is heated with concentrated sulfuric acid, often in the presence of catalyst like selenium, mercury or copper salts. These metals will help as a catalyst to break down the organic matters or break down the proteins, converting the nitrogen to ammonium sulfate. This ammonium sulfate in the second step, alkalization and distillation can be done. The digested solution is made alkaline now, typically with sodium hydroxide as shown in the picture, and releases now ammonia gas. The ammonia is then distilled and collected in a trap solution using boric acid. This ammonia gas is trapped by boric acid. Then titration can be carried out. The amount of ammonia is trapped in the trapped solution is determined by titrating with a standard acid like hydrochloric acid. The normality or the molarity should be standardized by using a primary standard like sodium carbonate. Then after standardizing you can find out the amount of standard hydrochloric acid used for neutralizing this base can be used for calculation amount of protein present. The volume of acid used to related to or the neutralizing to the amount of ammonia, and thus the nitrogen in the original sample is calculated. Now the second one, modified Damas method for quantitative analysis or estimation of protein. This picture shows the method how they did for modified Damas method. Here combustion reactor, reduction reactor, chemical water trapper, carbon dioxide absorber, and then comes TCD, that is thermal conductivity detector. Here mainly the thermal conducting detector you have to remember, which gives the detail about the quantitative analysis of protein. This method involves combustion, burning the sample in a high temperature furnace as shown in the picture, converting into nitrogen into various oxides, and then reduced these oxide to element nitrogen, which is quantified using, that element nitrogen is quantified by using thermal conductivity detector TCD. This method is alternative to the Kedal's method for protein analysis, offering potential advantages in terms of speed, safety and ease of automation. Now the third one is UV spectroscopy method for quantitative analysis of protein. UV, ultraviolet spectrophotometry, is a light spectrum. The light can be absorbed by any organic molecule, even protein. Quantifies the protein concentration by measuring the absorption or absorbance of UV light at 280 nanometer. This nanometer is specified by the lambda max, primarily due to the absorption of the light by aromatic amino acids like tryptophan and tyrosine within the protein. You can see the picture. The tryptophan, tyrosine, histidine, the lambda max, the blue color, green color and the orange color, you can see the peak of the tryptophan. They comes in the 280. Almost all the aromatic amino acids, they show the peak in 280 nanometer as taking as an absorption in the y-axis and the wavelength as an x-axis. You can see the lambda max by which you can find out. This is the instrument which is available in our lab, in our institution, which you are performing your experiments. You can see the graph or the computer which is shown. There is an uniform dots which is mentioned. That is a standard graph can be made by means of different concentration of the known protein. Absorption can be determined and an unknown concentration of the protein can be determined by extrapolating with the standard curve which is determined or performed. This method lies on the Beer-Lambert's law, which you might have studied in your analysis. Beer-Lambert's law. The light falls on a material. The absorption of the light depends upon the thickness of the medium and the concentration of the solution. So where the absorption is directly proportional to the protein concentration, providing the molecular extension. Molecular extension means exponential A value, coefficient, extinction coefficient E of the protein is known. So we can see that absorption can be used in UV spectroscopy to identify the quantitative estimation. Now we come to the example of gelatin. Repeatedly they have asked about protein, gelatin, because we are using more in the pharmaceutical industry and food industry. Synonym: gel form, purogel, gelatinum. Biological source: gelatin is a protein derived obtained by evaporating an aqueous extract made from bone, skins, tendons and other domestic animals. Some important sources are ox, Bostaurus, the cattle, and the sheep, Ovisaris, the small one, belonging to the family Bovidae. The gelatin preparation. The process of manufacturing of gelatin varies from factory to factory or manufacturers to manufacturers. However, the general outline of the process is given below. First one is raw material, bones, skin and tendons of bovidans or Bovidae family is collected and subjected to lining operation. Liming operation, not lining, liming operation. Lime, water. Liming process means in this process the skins and tendons are steeped for 15 to 20 or sometimes 40 days in a dilute milk of lime, calcium hydroxide in water. During this, fleshy matter gets dissolved. Chromoproteins of conjunctive tissues gets removed and fatty matters is saponified. The animal skin is further thoroughly washed in running water. Defatting. In case the bone and the material is properly ground or pulverized and defatted in close iron cylinders by treatment with organic solvents such as benzene. The mineral and the inorganic part of the bone is removed by treatment with hydrochloric acid. Extraction. The treated material from bones, skins, tendons is boiled with water in an open pans with perforated false bottom. This process can also carried out under reduced pressure. The clear liquid runs off again and again and is evaporated until it reaches to above 45% gelatin content. Now setting, the concentrated gelatin extract is transferred to shallow metal trays or trays with glass bottom. It is allowed to set as a semi-solid jelly. Now drying, the jelly is transferred to trays with a perforated wire netting bottom and passed through series of drying components of 30 to 60 degrees centigrade, increasing each time with 10 degrees centigrade. About a month is taken for complete drying. Bleaching is needed in case the dark color finished product is subjected to bleaching by sulfur dioxide. Bleaching offers a light colored gelatin as shown in the picture. Characteristic of gelatin occurs as a colorless or slightly yellow transparent brittle, partially odorless, tasteless sheet flakes and coarse granular powder. In water it swells and absorbs 5 to 10 times its weight of water to form a gel in solution below 35 to 40 degrees centigrade. It is insoluble in cold water and organic solvents, soluble in hot water, glycerol, acetic acid, and in amphoteric. In dry condition, it is stable in air, but when moist or in a solution, it is attacked by bacteria. Chemical constituents of gelatin. Gelatin consists of protein named gluten, which on hydrolysis gives a mixture of amino acid. Nutritional gelatin is an incomplete protein lacking tryptophan. The gelatinized compound is known as chondrin. That is the gelatinizing compound. Chondrin is the chemical responsible. And the adhesive nature in the gelatin is due to the presence of gluten. Detection of proteins and amino acid, and this test even for gelatin, common for amino acids and proteins also you have to write. Biuret test 1 drop of 2% copper sulfate solution with plus sample of gelatin if plus 1 ml of 95% ethanol, potassium hydroxide pellet if you add, a pink color solution in ethanolic layer is formed, indicates the presence of proteins. Million test, a drop of million reagent plus a sample of gelatin, a white precipitate indicates the presence of protein. Millions reagent means you should know that metal nitrate of mercury or mercuric nitrate that is dissolved in concentrated nitric acid. You should know what is millions reagent. Ninhydrin test, sample of gelatin with a few drops of 2% ninhydrin solution is then the test tube is kept in a hot water bath for approximately 5 minutes. After a few minutes, a blue color or a violet color indicates the presence of amino acid, amine and protein groups. Xanthoprotein test, a few drops of concentrated nitric acid plus sample gelatin A yellow colored solution indicates the presence of protein. For gelatin, on heating gelatin 1 gram with soda lime, smell of ammonia is produced. A solution of gelatin 0.5 grams in water 10 ml is precipitate, a white bluffy color precipitate. On addition, few drops of tannic acid, 10% tannic acid. With picric acid, gelatin forms yellow precipitate. These are all the chemical test for gelatin and as well as proteins commonly which you can specify. Uses of gelatin. Gelatin is used to prepare plastils, paste, suppositories, capsules, pill coatings, gelatin sponge, as suspending agent, tablet binder, coating agent, as stabilizer, thickener and texturizer in food. So it has a multi uses. It forms gelatinized gelatin with glycerin when it is added with glycerin, which is used as a vehicle for manufacturing of suppositories. Combined with zinc in the form of zinc gelatin, which is employed for tropical protectant, as a protectant. As a nutrient, gelatin is used commercially in food products, many, and bacteriology also they are used as a culture media. It has many applications. Now we are coming to the casein. Biological source of casein, usually it is from milk, but still you can see casein is a proteolytic enzyme. You have to remember in the lab which you do isolated casein from the milk. This is also important for you to specify in your record and as well as to revise your knowledge what you did in the lab, how you isolated casein from the milk. Casein is a proteolytic enzyme obtained from the stomach of cows. It is extracted from protein of the milk. In the milk, casein is structured in volumulous globules. Volumulous globules, that is why it is small globules. Emulsion. Milk is a natural emulsion which is white in color because all the light will get reflected due to this white small globules. These globules are mainly responsible for white in color of the light which scatters all the light rays, visible light rays. According to various species, the casein amount within the total protein of the milk varies. The casein content of the milk represents about 80% of the milk protein. The principal casein function are alpha S1 and the alpha S2 casein, beta casein and kappa casein. That is alpha 1, alpha 2 and beta casein and kappa casein. The distinguishing property of all casein is their low solubility at pH 4.6. Characteristic, the isoelectric point for the casein is 4.6. The purification protein in water is insoluble, while it is also insoluble in neutral salt solution. It is readily dispersible in dilute alkalis and in salt solutions such as sodium oxalate and sodium acetate. Casein does not coagulate on heating. It is precipitated by acid and by proteolytic enzyme. The name rennet proteolytic enzyme. You should remember, precipitated by acid, by a proteolytic enzyme also it can be precipitated. Chemical constituents, milk consists of 80% of milk proteins, casein. The major constituents of caseins are alpha 1 and alpha 2 caseins, beta caseins and kappa caseins. These caseins are conjugated proteins with phosphate groups which are esterified into serine residue. They have a low solubility at the isoelectric point of casein, pH 4.6. Uses of casein: it is used in the manufacturing of binder, additive, protective coatings, plastics such as knife handles and kitting, netting needles, fabric, food additives, and many other products. It is commonly used in bodybuilders as slow digesting source of amino acid. And now collagen is also another example for the protein products. Collagen is a protein. It can be considered a source of protein through it is not complete protein source. It is most abundant protein in the body, plays a crucial role in the structure of the various tissue including skin, bone and tendons. While collagen provides amino acid, it's low in the essential amino acid, especially tryptophan, making it an incomplete protein like gelatin. To prepare collagen for products, you can extract it from animal source like bones and skin. Benefits of collagen: skin health. Collagen is crucial for skin elasticity and structure, helping to prevent wrinkles and maintain healthy skin. Joints health. It contributes to the strength and integrity of joints, tendons and ligaments. Bone health. Collagen is involved in formation and maintenance of bones. This is about the packs, cosmetic use of collagen. Collagen can be used in skincare products like creams, serums and masks to improve the skin elasticity and hydration as shown in the picture. Kavach is another product which is available in the market by Ayurveda and herbal. This is a product which is produced by Arius herbal product. Kavach is often used to denote products like protein powders or herbal teas designed to boost immunity and overall well-being. Herbal and Ayurvedic ingredients. Kava is made of a blend of 13 herbs and plants known for their medicinal properties including fennel, red sandalwood, black cardamom and many. Immune boosters, they act as an immune boosters. It is marketed as a powerful immunity booster, potentially aiding a preventing various ailments including respiratory and digestive issues. Antioxidant rich content in Kavach. The formulation is designed to provide an antioxidant boost, helping to protect the body for oxidative stress and supporting overall health. Potential weight loss benefit is also there. Kavach is also promoted for its potential to support weight loss, possibly through its ability to improve digestion and metabolism. So this topic I have taken in such a way by discussing with the various question papers. So kindly Read, listen, prepare your own notes. And you are supposed to submit an assignment on the coming week without fail. I will inform you. Thank you. Thank you for listening. Subscribe my channel. Again I am repeating, and do the needful. Have a great day.